So my project is Genome Annotation for Community Curation. My name's Lily Andrews and my supervisor is Professor Mark Caddick. So outline of my research project. So first of all, I'll be going through the introduction to genome annotation. Next, I'll be going through my methods, which I use FungiDB, Galaxy, JBrowse, which are all the websites that I used. Results, I'll be looking at coverage plots, which I made in JBrowse. Discussion, I'll be overlaying what found in the results and what can be investigated next. And the conclusion is what we have learned about genome annotation. So first of all, what is genome annotation? Genome annotation identifies functional elements along the sequence of a genome. So essentially, on a DNA sequence, it's finding each region and giving function to it. So there's prokaryote and eukaryote annotation. With prokaryote annotation, there's 85 to 90% of the genome is coding. So essentially, it's a very simple genome structure compared to a eukaryote, which seems to be a bit harder to annotate the genome, as only 2% is coding. And there are a lot more complicated regions, such as splice sites, introns, exons, intergenic regions, all sorts. So it makes it a bit harder to annotate the genome. So there's two types of genome annotation. So there's manual and automated annotation. Manual annotation refers to a human physically doing the genome annotation themselves. Automated refers to a computer running the genome annotation. However, with automated annotation, there tends to be a lot more errors, as they might read two genes as one gene, or they might completely omit a gene altogether, which a human might see and realize the issue that's gone ahead. So RNA sequencing, this is kind of a new type of technology that's recently been able to make us see the whole transcriptome, as we can see both coding and non-coding RNA. So really, it's a really powerful piece of technology to give function to RNA. So it's been very useful for genome annotation. So fungi genomes. So the funding for fungi genomes, it's been quite far and few between. So for Aspergillus niger, there's only 30 labs that research into this fungi, whereas Saccharomyces cerevisiae, there's 1,800, which completely big difference, which shows that there's going to be fewer recent genome annotation or gene models for some fungi compared to others, which makes it a lot difficult if they use orthologs in genome annotation, that they rely on genome annotation which could potentially be wrong or old and not relevant. So one to note is Histoplasma capsultium. So I looked on FungiDB, and this genome this genome version hasn't been updated since 2005, yet this fungi exists as a dimorphic model, can exist in both yeast and hyphae form, as well as being a model potentially in disease pathogenesis, as it causes the histoplasmosis. So my aims of my project, I'll be looking at recent RNA sequencing data for fungi genomes with an older gene model. I'll then create a workflow on Galaxy to make this data in a more usable form, so then I can visualize it on JBrowse to compare the RNA sequencing data, which is more recent compared to the older gene model. I'll then code on Python to show this discordance, and then I hope to make this software free to use on GitHub. So my methods. First, I started with the data search. I set a criteria on FungiDB. As you can see, I used medium-sized genomes. I looked at which had RNA sequencing data and which had genome models before 2015, and it came up with these 5 fungi. And I looked at these 5 fungi individually, and I decided that Histoplasma capsultium had kind of a dimorphic model, and it could be a model for disease pathogenesis. So I decided this was best to work with, and it had a version in 2005. I searched Histoplasma capsultium and RNA sequencing data to find the RNA sequencing data from experiments, and I displayed this on Excel. I then uploaded this onto Galaxy and ran a fast QC to check the quality of each sample, and I separated each file based on its condition. In Galaxy, I then created a workflow to create this FASTA file into a bigwig file. As you can see, that's the workflow I used, and this downloaded onto FungiDB. On FungiDB, I opened the coverage plot of the bigwig file onto JBrowse. As you can see here, the blue coverage plots against a gene model. So in Python coding, I downloaded the gene model and the stranded RNA sequencing data, and I could see the discordance between the two data sets. So on GitHub, I plan to publish both the workflow on Galaxy and Python together on GitHub. So this is kind of showing that different people will then be able to see my published work on GitHub and use it for themselves. The results, as you can see here, this is coverage plot. You can see the introns don't align to where the coverage plot shows, which shows that the gene model might be a bit not as updated and may need to be changed. As you can see here as well, these two genes, they seem a lot closer together, or they may even be one whole gene together. But yeah, this is an issue that needs to be corrected. And here you can see the end of the gene needs to be extended a bit further along, as the coverage plot shows. So the discussion. So as I've shown with my results, there is discordance between the older gene model of Histoplasma capsultium and RNA sequencing stranded data. As I said before, automated annotation can lead to errors more than manual annotation, so it's recommended to use manual annotation. So next you could kind of carry out a study comparing Histoplasma capsultium in yeast versus hyphae form, which could better the gene model. You can also look at differentially expressed genes at different points in its life, as different genes are expressed throughout life. And differential splicing could be looked at, as differential splice sites occur in different conditions. So in my conclusions, as I said before, I've shown there's discordance between the older gene models and stranded RNA sequencing data in Histoplasma capsultium. Sufficient funding is needed towards fungi genomics to make sure that each fungi genome has an updated gene model. As I said before, there's a lot of differences between fungi genomes and how updated they are. So manual annotation of genomes should still be utilized, as automated annotation alone causes errors. So as there has been a data boom with a lot of genomes, automated annotation can be used, but it needs to be checked with manual annotation to ensure this is the top level updated data sets. It is important that the scientific community share information to keep genomic information relevant. So I feel like I've done that with creating this workflow that others can use, and it keeps an updated fungi genomes to help everyone. So I would like to thank Professor Mark Caddick and Dr. Evelyn Basenko and Dr. David Starnes for their help and contribution to this project. I'd like to thank you for listening, and here are my references. Okay, thanks.